I'm Ramesh Rao, a professor here in electrical and computer engineering, but for the last 12 years, I've been directing the UCSD division of this institute, the California Institute for Telecommunications and Information Technology, which is housed in Atkinson Hall, where we are gathered. By way of making a connection to the activities planned here today, I should mention that Cal IT2 has a collection of specialized spaces that has served us well in terms of bringing people together and launching new initiatives. And one of those investments is right here where you are. We have a collection of facilities in this wing, which basically speaks to visual analytics. How does the display of information in a visual format help engage and produce more insights than just working with numbers, for example? I'm hoping that we'll have some time, perhaps during the lunch break today, to give you some sense of the kinds of content and the kinds of experiences you can have when you're able to look at things at extremely high resolution. I should also mention that because of our interests in cyber infrastructure, we're also networked at very high speeds with practically anybody else who is networked at rates between 10 and 40 gigabits per second. We're just winding up a program called Cinegrid, which brings together digital Hollywood producers, equipment manufacturers, this whole new world where content will be digital from when it is created, to when it's viewed. So I think there's a lot of possibilities here, and as we look to our own future, I think we're eager to see how we can integrate it much more tightly into both research and the academic enterprise of the university courses, where students might get an opportunity to see things, not just read about them. So I'm going to be spending a good bit of time here hoping to learn things that might be the basis for new partnerships, and I would welcome any opportunities to have discussions and conversations with you along those lines. Thanks. Welcome.
Thanks a lot, Ramesh. That's very nice of you, and thank you for hosting us in this, what is, for those of you who haven't been here before, I think you'll find is a pretty amazing facility and something that we're very proud of here at UCSD. So my name's Phil Bourne, and I just want to thank you all for coming. I think we're actually trying to do something a little different here in this meeting, and already it's grossly different than usual. It's actually raining. I can't actually remember having a meeting here where it was raining, but anyway. So let me just sort of go into the sort of state of play. How do I change my own slides? Or?
Yeah, there's a keyboard in front of you.
Oh, yeah. Oh, keyboard in front of me. Thank you. All right, so I think what we're trying to do here, as I said, is something a little different. So let me just give you my little perspective on the state of play and what we might actually be able to try and accomplish at least. So I would argue that science is very inefficient. And I only speak from my own perspective, but Yolanda Gill, I don't think is here yet, who'll be here later. We've been working, oh, there she is, hello. We were working on effectively trying to create workflows around some of the work that we'd done only a year ago. And it was pretty hard to recreate what we'd done in our own lab a year later. And it's not that we couldn't do it, but it took quite a lot of time. And I've spent an inordinate amount of time. I'm old, and I think for 30-odd years I've been telling students they start off effectively doing many of the same things that others have done before them, including what I was doing when I was a graduate student. Now, there's good parts of that, but I think there are things that really could be improved. And that's something, and I think it's at all stages of the research pipeline, from the moment we have an idea to certainly writing grants to getting funding and how funding's distributed and all of that. And so, in fact, Peter McCartney from the NSF, who was kind enough to fund this initiative, is here, and hopefully he'll be active in the dialogue where we talk about these things, because how we fund things is definitely part of this equation. So we're here, of course, because we all do e-science of some form or another, and there's no question that that's had profound impact on science itself. But in some ways, it's also contributes to the problem. An example of that would be that if you go and you try and find software to do a given task, you can spend an inordinate amount of time trying software from various places, if you can get hold of it, We've actually looked at this in a more quantitative way, and it's actually quite depressing in terms of significant resources went into making software. And yes, GitHub and all these other resources are clearly important developments, but in many cases, it's an inefficient process. And so I think the idea, and that can apply to data and methods and methodologies and a whole series of other things. So I think we can potentially do better there. So I'd also say that the reward systems and that relate to contributions are not very well correlated at this point in time. There's a simple example of that would be, what's more valuable, a paper that's published and is only cited by the people who wrote it, or a data set that's downloaded and led to 100 papers? I mean, it's obvious, and yet there is effectively no reward for generating that data set right now. And that's another sort of an example of where the reward system, which is critical to any kind of change, comes into play. Resources themselves are siloed, and I'll give you examples from our own experience about that in a second. And I think they've served us enormously well. I think, particularly in the biomedical sciences, we have some absolutely amazing resources that have enabled us to go to do science in incredible ways, and just the access of those resources illustrates that. But it's got to the point where we tend to ask questions that almost invariably can't be answered by using one of those resources alone. That, in effect, the silos are, in my opinion, becoming somewhat limiting. And so the idea that we begin to address that issue is another idea that comes from this. And I also say that it's clear that different types of social networking are having a large impact all over the place, but it's not clear that they're having the kind of impact on science that we would like to see. Certainly there are things that are emerging. I think ResearchGate, Biomed Experts, and Mendeley are examples of things that maybe the older people here may not be using, but that certainly people in the labs use. And there's the notion of the beginnings of community around those kinds of resources. And that's sort of an underlying theme that I would hope we could bring out in this meeting, is taking that farther, and you'll see what I mean in a minute. So let me just sort of illustrate this current state of play with something that I'm involved with, as a number of people in the audience, and that's the RCSB Protein Data Bank, which I'm sure most of you know, if everyone knows, is a very well-used resource. It is featured in National Academy reports and other places as an exemplar for what biological resources should be like. And what I'm about to say is that in some ways that's true, but I think I'm going to use it as an example for how we could go so much further. So an example of that, of course, is the paper that relates to the PDB is one of the most cited papers in all of biology. It's had over 14,000 citations, but no one has ever read it. I mean, there's no reason for reading it. It purely is there to provide some level of reward about the data that's in that resource. And, so in some senses it doesn't make sense, but it's the only metric that we have currently for actually showing value. So the idea that we might bring forth different types of metrics is something to think about in the context of this meeting. I would say that, and this sort of relates to the siloing nature, is that, from the kind of work that we do, and I'm speaking somewhat personally here, I mean, we would say that structure is the devil in the details of systems biology. But in the PDB itself, at this point in time at least, says very little about systems biology. It's not, it's really a retrieval point. And I would say that it's built half a community, and let me explain what I mean by that, is that where I think it's been very good at building community is among the people who deposit into that resource. They feel like they have shared ownership, and we, in maintaining it, are just custodians of their output. And but they trust us to do that, and part of that trust comes from the idea that we we work with them at every stage of the way. So, for example, if we're concerned, as we have been, about the quality of data and we want to improve validation, then the community is brought in through task forces and other means to actually define what it actually means to validate a structure. And then that takes place. And so it's actually driven by the community. So all of that's a good thing. So why do I say we're half of the community? So this data comes, and then what happens to it? It sits there on a resource, and it's used by 300,000 unique people a month. It's not that it's not used. It's used a lot. But it's not in of itself really built community around the users of that data at that point in time. Yes, we do what we can to achieve that by having workshops, going to meetings and talking to people and so on, but it's not a collective. It's not where people can come and, for example, plug in their own applications around that resource. And so you could imagine the notion of it being something like an app store, where people actually deposit apps. There's a certain quality control around that, and then the user determines what it is that they want to do with that resource, and which of those applications they want to run, just as you do on your own phone. And then you run that. If you don't like it, you try another one. And then there's a dialogue that goes on between various people associated with using those apps. So it's the very early beginnings of the idea of doing community. And so that is really the sort of theme, because there's a whole group of us here from quite different and diverse backgrounds. So the question is, can we come together to do something like this? So can we do better? So the sort of underlying question that we'll be probing in the next 2 days is, how do we establish a vibrant, open and collaborative community to support this kind of simulation? And I'm just interested in facilitating this. This is not my personal area of research, but I think it just strikes me that something that can be improved, and there's lots of incredible things going on already, of course, and we'll be hearing about those. So that's really the question. So what's the conceptual approach? It's basically asking ourselves the questions, what can we hope to achieve in the next 5 years? What are the impediments? And then how can we get there? And this is something that in each of the speakers will try and address in some way based on their own experiences. Pragmatically, how's this going to work? It's going to be a little different. The speakers we're going to hear from a whole diverse and amazing set of speakers, but they only get 10 minutes. And because of the nature of the numbers and so forth, we really are going to have to stick very closely to that. And so, speakers have been asked to really be succinct and give TED-like talks. Some people knew what TED-like talks were, but we'll see how well it pans out. But the idea is just to get key points across, and particularly around these conceptual questions. We will be capturing, what is said here, both in video. There's a video feed now, and already it's clear that there's quite a lot of people, probably more than that's in this audience, who are watching the video stream. There's also Twitter feeds and other commentary mechanisms that people have been fairly active on already. So we'll be gathering a lot of what is said at this meeting. And we're also, capturing concepts. So Sophia and Wynne are doing this already, so you can see that. I've used this in different contexts, particularly when I was working with the Gordon Betty Moore Foundation, and I found this to be an amazing way to summarize what happens in a meeting. And they're going to be doing this throughout. These will be the pictures of these concepts will be tweeted. And also they'll be available for you to see. They're also you're not just passively looking at these things, you're an active participant. So the idea is when at the breaks or any other time, you can go up to one of them and essentially say, I have this idea, and they will, or thought, whatever it is that relates to what you think is important to get across, and they will conceptualize that on what I don't know what to call them, the concept boards. And you'll be able to see those throughout the meeting. Then another approach to this is that if a speaker speaks for less than 10 minutes, then we could use that time for a question or two. But other than that, after a group of speakers have spoken about a particular topic, we will then have essentially a panel discussion, which will be moderated by someone. And the idea then is to sort of bring out the key things that it could be a pressing question for a given speaker, but more than, ideally it would be some underlying theme or some underlying concept that comes from at least two or more speakers that we really want to begin discussing. And it's really just finding out what those things are that are worth discussing amongst ourselves is really the purpose of the talks. Then there'll be what we call an open space session this afternoon, where there will be a series of tables where there are topics. And you just go to one of these tables to discuss a topic. Those topics will be defined by what comes out of this meeting this morning and up until early afternoon. And then there will be people who are assigned to those open space tables who will gather information. And then tomorrow we'll have some more talks, and then we'll be then drilling down on some of the concepts that have clearly stuck to effectively define working groups, and hopefully they'll be self-defining. This is, let's say, quite an experiment. That people will essentially appear out of the woodwork, so to speak, to take an active role in all of this, and we'll form working groups to essentially build on some of the ideas that have come out of this, as if we're going to have some kind of central entity that is going to try and help build a community where, yes, it will have software, yes, it will have data and a whole series of other things, but what should it really look like? You're going to define that, or help us define it. So the end product, it's not actually a secret agenda, but it's, this is my way of saying, why should I write a grant? Let me get you to write the grant. So we're kind of crowdsourcing what I would hope would ultimately be something that we could call the next generation biological collaborative. So we'll see how well that goes. So just let me remind you of what the question is that we're sort of trying to address overall here. And I guess we can think about moving on. I guess, is there any absolute pressing question that we need to address? I should say, because of the webcasting, we need to use the microphones for the questions, and there's supposed to be someone wandering around. Oh, thank you. Doing that. And then there's things like putting your cell phones on mute and that kind of stuff, and don't listen to the live feed when you're following the audience, or don't listen to a previous presentation when, not without headphones anyway. So is there any pressing question before we get into this? Mark.
So the last word, simulation. Can you comment on why you're limiting construction of virtual cells to projecting them into a simulation space?
I wouldn't say we're restricting it. I mean, in the sense that what's driving this, the underpinnings of this, is the notion of a software center. And that software could be used to run simulations, but it could be used to do a lot of other things around whole cell modeling as well. So yeah, perhaps that's a bit of a misdemeanor. So certainly one of the things we can do here is to redefine the question. I mean, there's nothing wrong with that. And there are no rules here. And for people like Mark, that's perfect. So that's the rule. The golden rule is there are no golden rules. George Bernard Shaw.
Can you hear me? Okay. So what is the purpose of constructing a 3D virtual cell? Are we using this for anything predictive, or just to, try to collaborate, cross-collaboration? What is the actual goal in mind long-term?
I mean, from my point of view, and again, you're going to define the goal to some extent, but it's really a gathering point of infrastructure and people to achieve things in a way that is accelerated, that would, the sum of the parts is supposed to be greater than the whole. Can we do something that we would not have been able to do otherwise? Can we accelerate the rate of discovery? Can we change the way we think about some aspects of the science itself, or the reward system, or the funding mechanism? At the very least, make the points of what should happen. Okay, I mean, there'll be plenty of time to discuss all of this, so let's move on. And our first speaker, in deference to the fact that they only have 10 minutes, we will not do elaborate introductions about people. It's all there. You can pull up your laptop and you can see who they are. They're all amazing people, and so we won't spend time with introductions. So our first speaker is Andrew McCulloch.